and go for it.
Oh, yeah. Okay. Yeah. So my name is Veerle, and I am I'm a professor at the University of Leuven, and that's in Belgium. And yes, I like in 2015 already. We encountered, let's say, the first. It was a PPP2R5D patient, and I got involved in the research from the very beginning. So I was contacted by a Norwegian genetician who diagnosed, I think the very first case, it was Regina was her name. Maybe some of the people here know Jannike, the mother. She was also in the Boston meeting. And also I never met her until that time in Boston. So I mean, it was very nice to get to know her. But I have been working on the PPP2 genes and proteins for my whole scientific career, let's say. So ever since I was a PhD student, I worked on several aspects of these genes and these proteins. So that's why this Norwegian genetician also contacted us to help him figure out whether these mutations were actually pathogenic or not. And so the whole thing went to roll, and it kind of caught me, or I don't know how I have to explain in English, but I mean, having met the families and how grateful everybody is to, I mean, that there is, I mean, a research team looking to help. I mean, I find it very rewarding to do what we do, and very motivating also, that we can hopefully make a difference for everyone. So I said, ever since 2015, I said, okay, I'm gonna do this till the end of my career, and hopefully we can do something to help to understand how these mutations work, how they cause the disease, and from that type of understanding, maybe also try to see whether we can find some drugs that could help to interfere and improve the condition, or maybe cure the condition. But it's of course a long road, because 10 years ago this disorder was not even known. So since then, I think we have come already a long way, and we have progressed a lot, but there is still a lot that we don't know. So research is, I mean, it is still needed. And okay, so we engaged ourselves to this effort, and yeah, we're doing our utmost to, let's say, to move things forward. And yeah, so I'm also very grateful for JGA. I think, yeah, the fact that was also really a coincidence, and that they managed to support the research in such an amazing way and still managed to do so. I think that has also been the cause that the research has moved on so well. So also very grateful for that support. Without that support, I don't think we would have, or we would stand where we are now in terms of the research, by no means, because yeah, research, it is expensive. It's not very fast, although we make progress every day. It's very small steps, and sometimes I realize that it's maybe too slow for many of you, but yeah, that's what it is. We cannot really change that too much. It is inherently inherent to the nature of what we do, that it is not so fast. But yeah, we're doing our best. So, and yes, I think I've always engaged very well with the patient community, and I hope to keep doing so, because it is what motivates us, really. And I really enjoyed meeting also everyone at the meetings. I went also to Germany last summer. I found that also very, rewarding to get in contact, in close contact with the people, and also try to explain as to the best of my abilities what is going on, because that is also a challenge sometimes. If you are not scientifically, educated, like how to explain what it is all about. So maybe a too long introduction, but and so for today, I'm not really sure. I wasn't really sure what was really expected from me. I prepared some slides on the paper. So the paper was submitted in August, and I'll explain what is in the paper. So this is actually what I prepared. It is not yet accepted. It should have been last week, but on Wednesday we got the message from the journal that it was not good enough yet for acceptance, so that it needed another round of revision. It's stupid because it is all about a statistical test that the reviewer didn't agree upon, which doesn't change anything to the message of the paper, but the editor followed this reviewer and he still rejected the paper, but we're allowed another revision. So next week, I have an appointment with a statistician to solve this question. I mean, we use the same kind of statistic method for all the papers that we published on these syndromes, and was never a problem, and now all of a sudden it is a problem. So the paper was submitted in August. We got back the first decision in September. We revised the paper, submitted it back in October, and now last Wednesday we got the decision that we need another round of revisions. So I think it will probably then still, if all goes well, we can submit after Thursday again, but then it will have to go back to the reviewer, have to undergo another round. I mean, he still has to give his consent, and then hopefully in maybe 3 weeks, I guess, hopefully it could be finally accepted. But maybe I don't know. Is everybody okay with me presenting a little bit further, or do we want to talk about other things, or
I think they'll be
how
helpful,
do you
Verly,
see?
if you
Yeah,
do a few
yeah.
slides. Do you all agree? And
Um,
then we can open it up for questions. Maybe just, keep
yeah, okay.
some time for questions as well.
So let me see if I can, how do I have to do this? Share my screen, I guess. This one. So can everybody, hang on, can everybody see this? I'm gonna. Okay, so does everybody see this?
Yes,
I don't
can.
hear
Yeah.
anything.
Yes.
Okay,
Yeah.
okay. Yeah, so
And
this is what
if you have any questions throughout, feel free to, throw
yeah,
them in the
just,
chat, or just, speak up, whatever works better for
yeah.
you. And it
So,
looks like we have one more family joining, but you can go
okay,
ahead and then we'll do the introduction later.
yeah, okay. So do feel free to interrupt or ask questions whenever you think you need to. So this is the history that I was saying. So we submitted a paper to American Journal of Human Genetics. The good thing about this is that it is really one of the top journals in the human genetics field. So if it will be accepted, it means that the paper will have a very, very broad exposure, every human geneticist reads the American Journal of Human Genetics. So I think this will also mean that once the paper is out, I would predict that we will see more diagnoses pretty soon, if they are out there, of course. So it was a huge effort. So there was a number mentioned of 30 patients, but at some point we had actually 36 potential cases. That was the maximum number. In the meantime, I have in October, and then 2 weeks ago, there was another French case and a potential other French case and a potential other Swiss case. People who contacted me that are not in the paper because, yeah, that was too late. So let's say potentially 38. But when we started to look at the variants and whether they were pathogenic or not, when we started to collect the clinical data, there were four cases that the clinicians never responded to us. So the cases were reported in databases as potential pathogenic cases, but yeah, when we contacted the clinicians, they never replied. So we couldn't provide the clinical data for that reason, and so we had to leave them out. And for the remaining 32 cases, then we only found evidence based on our assays that we do in the lab of the fact that a variant that was reported that was indeed pathogenic for 27 cases. So 27 is the number of cases that we report in the paper, of which we are sure that the genetic mutation that these children have, that it is the cause of this disorder, let's say. So there is 5 others, and I think some of them might actually also be in the Facebook group, for which we are still not sure that it is a pathogenic alteration or not. I would even say we rather think that this alteration is actually not pathogenic and not causing the disorder. So yeah, so this is the mail I was mentioning. So it says, so it was sent this week. So this should have been the accept, but it was not. So it says here, reviewer 2 has some remaining concerns about the statistical analysis, blah, 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 blah. So we have to answer him and submit a revised version, and we have 4 weeks to do this, but we will do it sooner. So probably end of next week, if the statistician has a solution for our problem, then we can resubmit, and then hopefully I don't expect it to be problematic. It should be accepted then afterwards. But that might still take then, 2 weeks or so before the journal really decides that it is accepted. But normally, let's say it will still be published this year. So that is the good news.
It'll be your holiday gift to all the families.
Yeah. But it's also a relief for us because it's like a long time already in our minds and in our hands, and I mean it would be really nice if it is out. So maybe just I go one back here and pay proper credit to the person who really, I mean, moved all things forward, and that is Iris. I think some of you might already also have met her in Boston. So that's a postdoc in my lab. And yeah, she actually did all the biochemical data experiments and the coordination between, these 60 or 70 authors. And I can tell you it is a real pain, if you have to get all the relevant information from all these single persons, and you have only a subset that provide you the data right away, but there is always some that you have to chase 4, 5, 6 times before you get all the relevant information. So it has not been very easy. So many credits to Iris. And then also Sophia, she's a Greek clinician, and she helped us to analyze the clinic and to describe and analyze the clinical data. Because we are not MDs, we are no medical doctors. So for us, it's a little bit more difficult to do that. Okay. My computer gets stuck. Yeah, here we are. Okay. So just, yeah, so as I said, 27 individuals with proven pathogenic variants. So the oldest was 29 years old, the youngest 14 months. And for the sake of Rachel, there were 16 boys and 11 girls in the whole population, and 20 different mutations, 20 different variants. So some of the families provided consent to publish photographs. So I see Alan here and Ahmed. Elena is also there, I think. And then Alfie, I'm not sure whether he is there, but it's I mean that doesn't really matter. He's probably in the paper anyway, but without a photograph then probably.
Yes, sorry. Yeah, no, I can't see his picture there. I think we did consent to photos being used, and I know our geneticist did take some, but maybe you never got them. But yes,
no, but
yeah.
but it's okay. I mean, it's not a problem or anything. But so based on, I think it's like 10 pictures here. We got a computer, so artificial intelligence generate like a general image, and that is presented here on the right. I mean, that is often how clinicians, so based on some facial characteristics, clinicians sometimes can already, how shall I say, predict like what type of genes could be affected if they are very good and some are very skilled in this. So this is really, for the clinicians, it is important to have this. So that's why we did it. It's the first PPP2 paper where it has been done for. And so this face here is a computer generated face. It's almost unbelievable, but and this face is generated based on the characteristic that we see in these 10 the patients over here. So that is one of the figures in the paper. Then this is a list of, it's very small, I apologize, but so here you see the 27 patients with the variants, and then the clinical characteristics. But I mean, I don't have time. I mean, I cannot go through this, but I have some summaries of this later on. So these are the 5 individuals with specific variants and even a full deletion of the gene, of which we think that these variations are not pathogenic. So they may not be completely neutral, but they do not give rise to the typical symptoms that we associate with the PPP2R5C condition. And as you see, some of these variants are also inherited in this case and in this case from a mother that is asymptomatic, that doesn't have any symptoms. So this already also indicates that this alteration, a complete deletion, that it is not really pathogenic. So these, we have reported them in the paper, but we explain in the paper that we don't think that these are pathogenic alterations. Then this is an overview, let's say, of so maybe I go back one slide. So this is already a very important conclusion, let's say, from the or a very important impact, kind of impact of the paper of a paper like this. That is that we provide a clear diagnosis to those 27 individuals in the list. It is a firm confirmation of the diagnosis that is important, and an establishment of PPP2R5C as a new genetic condition, let's say. So I think that could be important for some of you, to get access to all kinds of support from the government or whatever. So that is a very important first conclusion. For these 5 cases here, then it means that this diagnosis that they might have got might be not the right diagnosis. So positive and negative things, let's say. So the second reason why, of course, this paper is important and to add as many patients as possible as well is that it allows us to describe the PPP2R5C condition better. And so if we look at the developmental delay, and that can be measured, by motoric development of the motoric abilities or the language, the IQ, that kind of things. So that was reported in our clinical table. So then these are the figures that we get. So with a developmental delay let's say that is mild in about 50%, and then 20% more severe, 33% more moderate. I have to say, when we compare these figures with, for instance, the PPP2R5D condition and what we know about that in general, we have to conclude that PPP2R5C is actually seem to be milder affected. It's a milder condition. Of course, it's general speaking. It's a generalization, but overall, it seems milder than R5D. So these are like parameters, growth parameters. So height, weight, and OFC. It is a difficult name to measure the head circumference. And this is a very important conclusion. So most of the children have larger heads, bigger heads, and above a standard deviation of 2.5. So that is the darker blue. Then the deviation of the head circumference has become that big that we speak of macrocephaly. So that is the scientific term to say that the head circumference is unusually large. And this is also something that we see often see occurring in the R5D families. So height and weight are more in the normal range. Then most of the cases had neonatal concerns, so 3/4, and it could be different things. So the macrocephaly is also here, but most pronounced is the hypotonia. So in 21 of 26 cases, it was reported. And this is likely also, it is something that also lasts. It is lasting through life. It is already noted, so let's say right after birth, but it doesn't go away. It might improve a little bit, but it usually never goes away entirely. And that is also linked to several additional problems that I will show later, like eye problems or gastrointestinal problems that all have to do. So the hypotonia is a difficult word for low muscle tone. So that affects the gastrointestinal system, for instance, or the eyes sometimes. Yeah, feeding difficulties, respiratory support. Yeah, that was what was reported. Then let's say 13 out of 24 cases that we had data from, we reported behavior problems. It's a very broad term. Yeah, autistic traits was most frequently reported amongst those problems, but also like stereotypic behaviors or hyperactivity were also amongst the reported ones. Then something that is of importance, I think, is the frequency of epilepsy, so occurrence of seizures. It was a high percentage, so more than 50%, 54% of the cases it was reported. What I added some more information here. So the mean onset of the seizures occurred at age 4.2 years. But and 50% of the seizure-free individuals were younger than that age. So it means, yeah, it could be the percentage could become higher when the children grow up. But there were also 2 cases that were older, like 14 years, 29 years, and that never reported any seizures so far. So the type of seizures is very diverse. So there is not something that you say it is always this or it is always that. So it ranges from absences, which are just very mild, to status epilepticus, which is very severe, and a relatively high number of refractory epilepsies. So when we compare this with R5D, I think we have to conclude that it's probably a little bit more frequent than in the R5D population. And then the last thing that we systematically, let's say, questioned is the brain MRI. So if there is by scanning, if the brain looks normal or abnormal, and yeah, there were also here in 59% of the cases some abnormalities were reported, but here again nothing that was always recurrent, and no real developmental problems or anything. Everything was usually very well developed, but yeah, there were some issues sometimes, like bigger ventricles or some white or gray matter issues in some cases here or there. But this is also very similar to what we see in R5D. So it is the 2 other genes, the CA and the R1A genes, they have more brain abnormalities when you look at brain MRIs. Then the comorbidities that we found, so this means like associated issues that might be related, might not be related to the condition, were, as I mentioned, GI system issues, going from constipation to diarrhea, but that likely linked to the to the hypotonia, so the low muscle tone, sensory organ issues, usually the eyes, but in a couple of cases also the hearing, and endocrine issues were reported in 3 cases. So that went from early puberty, there were type 2 diabetes, and also some calcification leading to kidney stones, for instance. That could also have an endocrine origin, but it was not something that was, recurrently reported, let's say. So not sure whether it is, by coincidence related or whether it's really related. Yeah, we need to have more cases to make that as a more firm conclusion, let's say. Okay.
Really, I have a
Yeah.
question real quick. Have you seen, and if you're gonna capture that later, just tell me, I'll capture it later. But have you seen any trends when it comes to like the head size and the severity of the symptoms, or like different variants of
No,
anything?
yeah. So
Okay.
I mean, I was gonna touch upon that when discussing the variants, but it's different. It's a little bit different than for R5D. Uh, so there is no real, you know that very well, Carol. For R5D, we defined like these three groups, severity groups. Like two more severe groups and then one less severe group, and that correlated very well with the biochemical impairments that we saw of the variants. Here, it is, we can actually only see like a group 2 and a group 3. And the correlation with the severity, it is all not so clear. So it is less clear than for R5D. But maybe that is also because it is overall milder. But I think as a general conclusion, and I go to the next slide here, that is that the variants that are occurring here in this region of the protein, so this is like the R5C protein that I show here with the numbering of the amino acids in the protein, so the building blocks in the protein that all get a number. And so usually all the variants are missense variants. It means that one building block, the right one, is replaced by a wrong one. So an example is like an E is changed into a K. A red is changed into a blue building block, let's say. And so the changes occur in a clustered way. So there is a cluster here, there is a cluster here, 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 and here. And we know, we know the crystal structure of the protein, so we know where these variants actually occur in the protein. And what we see also that in R5D, that the most mutations occur here in this orange cluster. And we call this the acidic loop. It is a loop structure, and there we see the most severe variants. And it is also like that here for R5C. So here it's more severe. When we go to this region or here and here, it seems less severe, and the green, it's more variable. But the link with the biochemical impairments, it goes everywhere. It goes into all directions, and there is not really a logic to this. So it's less clear than for R5C, like how the severity links to the biochemical impairment in the variant, or even how severity can be linked to a variant. It's more diverse. Of course, it's only 26 cases, and so it could become maybe clearer when we would have more cases, but I'm doubting a little bit about that. We would have seen it. I mean, 26, it's not a lot, but it's not little either. So we would have seen it, I think, if it were like that. So it's less clear than for R5D.
And are there any variants that seem to be more reoccurring?
Yeah. So wait, maybe I have to go here. So this is it looks complicated, but I'll try to guide you through this. So this is like yeah, here I make a kind of an alignment between gamma and delta. So B56 gamma, that is the protein name, that the name of the protein that is encoded by R5C. And delta, B56 delta, is the name of the protein that is encoded by R5D. So the upper lane is the sequence of building blocks, let's say, that as it occurs in R5C, and below is as it occurs in R5D. And this is all the sequence of, and if I go back here, of all these conserved regions here that are affected by the mutation. And if you look in detail to, the green, the yellow, the purple, and so on, and to these sequences, you will immediately notice that the R5C and the R5D sequences are completely 100% identical. So it shows you that R5C and R5D, they are highly related. They are highly related. And the proteins are also affected sometimes in the same residues, sometimes in different residues. And this is what I try to show here. So if we go back here, in this first part of the protein here, the green part, so this is the sequence. So above is the R5C sequence, below is the R5D sequence. They're identical, and indicated in red are the residues that are mutated. So for which we find cases with mutations in those specific residues. So you see in this region, we only see mutations in the R5C. And the same occurs here in the 8 loop. Also, only red variants or red residues in the upper lane, so in the R5C. So it's represented here too. So in R5D, we don't know any patients that are mutated in those regions. So that is a difference. And maybe also to show you here, so in gray here, these gray and these helices, that is the structure of the R5C protein. And here is another protein that is part of the PP2A complex. It is the catalytic subunit that is actually the protein that has the biological activity. But maybe I have time to explain that later on. But so you see like a helix, and then there is a loop, and then another helix. And the loop, that is what's conserved, and all the loops are kind of facing towards this brown protein here. And it is usually the loops that are affected by the mutations. So it's like that in R5D, and it's like that in R5C. But so for R5C, coming back to what I said, only loop 1 and 8, they are affected. R5D, we don't have any variants in that. The loop that is the most recurrently affected, both in R5C and R5D, that is this one, the orange one. So if I go back here, this one here, there we see the most mutants, and the majority of those mutations give rise to usually more severe phenotypes. Not all of them, because there is also milder variants there, but difficult to generalize. So this loop, yeah, it is this one here. It's a very long one. You see, it's a little bit, the sequence is a little bit longer. And so it is upper one again. These are the affected residues in R5C, and below are the affected residues that we know in R5D. wait, so I go here. So this is the acidic loop. We call it the acidic loop. So we see some variants, the same amino acids, so the same building blocks are affected, like this E and this E and this E and this E and this W and this W. But you also see that there are changes. So some variants are only present in R5C and some are present in R5D. And I also have to say, so if the building block is affected in R5C and R5D, that the missense mutations are also identical. So for R5D, we know, for instance, that E198K, so that is here, that has the highest occurrence, the highest recurrence. So 60% of the reported cases in the literature have E198K substitution. So the same residue in R5C can also be affected. Then it has a different number, E122, but if it also is affected by a K mutation. So E122K and E198K, it's the same mutation affecting the same region in R5D and R5C. For R5D, we know that it is usually associated with a more severe phenotype, and also with R5C, it seems to go into that direction. But it is not the most recurrent variant there in the 26 or the 27 cases. The most recurrent mutation occurs at the position H133, that is here, and we have no patient whatsoever that is affected in that variant at that position in R5D. So you see there are similarities, but there are also differences. And it probably relates to slightly different structure of R5D and R5C, but so we don't know yet what it means, but obviously this loop is important for the function somehow. We don't really entirely understand completely yet why or how, although it affects the binding of this C subunit. But yeah, I lost it.
Yeah.
I lost it. So I mean, yeah, similarities, but also differences. And so if we would have so H133, we have 5 patients. I think that I have to see. Yeah, so we had 5 patients that were affected in that residue, and E122, 4. And then 2 here at the A and the W, and then all the rest were kind of singletons. So
Girly,
yeah.
if you don't mind, let's pause for just a minute. I know some of this is a lot of science, and don't feel like you have to understand it all, and it's okay if you don't,
No, I just want to, by showing this, I just want to show that, yeah, that R5D and R5C, that they have a very similar structure, and that often the same building blocks are affected and by the same mutations.
Yeah.
And
So a follow-up question on that, and I know that might be on the family's mind. What does that tell us in terms like now we're approaching like clinical trials for one of the drugs for
yeah.
2R5D? Does that tell us that we could potentially have the same successful results? Assuming we have successful results, we haven't really, we'll see. But does that tell us that we could use that same learning for 2R5D as well, 2R5C as well?
Yeah, I cannot answer that question, but it is possible, of course. It's logically speaking, yeah, there is a strong relationship between them. The 2 proteins might share similar functions. They probably also have some different functions as well. But it makes it quite likely that I mean, if we would have to include patients that are affected in another gene than R5D in the R5D clinical studies, then it would have be these ones. Then it would be the R5C. They are by far the most likely affected than R5D. But I cannot positively affirm that because we have not studied. We have not studied any R5C models so far. And we have not even made any R5C, let's say, disease models so far, where we first should test this, of course, before we come to that kind of conclusions. But if there is any gene that could benefit from the same kind of treatments then would be found, then those that would be found for R5D, it would be the R5C individuals, for sure.
Okay, great. Thank you. Any questions from the families on variants or the symptoms or anything else before we move on with the presentation? And Simona, welcome.
Hi, hello. I have, thank you for your presentation. I have just, it's not a question, just a comment. When we were invited to send the data, I'm speaking about Nicolò. Nicolò was very little, and it's usually that every paper takes time to be written, to be prepared, written, and everything. And when we completed the database, Nico had some situation that for sure changed during the time. At that time, for example, Nico didn't suffer of epilepsy, and now it is. So I was wondering if it's too late to give this information because already the paper is submitted or not.
Yeah.
Also the behavior, there were some, when we completed, Nico was 2 years old, and some typical behavior that now he has at that moment were not present. So just to be more complete, in the description of the cases.
Yeah, I don't think we can change anything to
No, too late.
anymore. But of course, yeah, the description that you provided, it's always related to the age. So that
Yes.
and at that age, the description was accurate, right? You see what I mean? So what
Yes, it was related at the age.
yeah,
Yes.
at that age, it was
Anyway,
accurate
yes.
description. So yeah, I don't think yeah. As I mentioned, I don't know. You arrived later, eh, Simon? I
Yes,
don't
I
know
know.
when you arrived,
But anyway,
but the paper is
for example,
submitted.
Nicole,
It's
yes,
almost
submitted.
accepted.
No, it was a pity. Maybe, yes, we could update the data before, yes. But
I
anyway,
was
he suffered.
Well, just a quick comment on that, Simona and others. Like one place to keep the data up to date is Simon Searchlight. Like we can spit out a publication
Yes,
every 6
sure.
months, but we can
We
have
did,
these reports
yes,
quarterly. So just make sure, and that's gonna help us, see how as our
yes.
population ages, what that looks like. So just make sure you
Yeah,
stay up
yes.
to date on these within Simon Searchlight.
In Simos, we updated these
Perfect.
all this
Yeah.
information. But just a question, because 27 cases are a lot, no? Because I didn't know so many cases, but all these cases are we know if are also present in Simons or not. With this, we cannot know.
They're not. We only have 8 in Simons.
Yeah, but
I know.
I'm gonna do a big effort to motivate now the clinicians to yeah, to get it back in touch when the paper is accepted,
Yes,
to get back in touch
yes,
with the families. And then, I mean, I mentioned already to some of them, I mentioned JGA and so on, but yeah, sometimes the information doesn't get through. Sometimes
yes,
I don't even, I am not in touch with the clinicians, but it goes through some consortium or a
yes.
database or so. But I mean, once a paper is accepted, I have to send
Maybe.
everyone a message like the paper is accepted, and then I will convey in that same mail, I will convey the message like, look, you have a diagnosis now
Yes,
for your patient. Please mention mention it to the families and
Yeah.
mention JGA, mention there is a support group,
yes,
and I will just
yes.
encourage them to
Uh,
do that. And then we
Verily,
will see what will
Sharon,
happen.
Sharon and I will be working on like a 2R5C specific welcome letter. So let us
Okay.
create that for you. Then you can just attach it with this email, and it'll have all that information in there about like the group,
Okay. But
Simon
then
Searchlight. It's so
you need to
we'll
be quick.
it'll be quick, I promise. So I'll take
Okay.
that action, and
Oh,
we'll
but
collaborate to create that.
yeah, but
Okay.
that would be great because sometimes, yeah, like I said, we started from 36, and but then by carefully looking at the data, by doing all the biochemical assays of the variants, we only end up with 27 that we are really sure for which to which the diagnosis applies. So that's why I always am also careful to say, okay, this is the group you belong to before you will really have the data that confirms that this is really the right group that they belong to. So I think once a paper is accepted, we can nicely, I mean, confidently do so and spread the message and spread the word.
Can I
Um,
ask a question too?
yeah,
So
sure.
of the 30 some families that you have that you know of, are these like specific geneticists that know to send their family information to you, or do you have access to anyone who's run a genetic report that a C has popped up on? Like, could there still be C's floating out there that were sent home with the letter that said it was insignificant or of unknown significance?
Yes, that's what it all says now, I think.
Okay.
Um, because this gene is not out yet as a real disease gene. So that's what the paper will change. And that's why I anticipate that I will get, once the paper is published, we will get more cases and we will get more mails to say like, oh, I have a case and the variant is not in the paper. And can you please test the variant? And it went like that with R1A, with R5D as well. I mean, at some point you have an explosion of diagnosis based on once this is really out in the literature and people put it on gene panels and that is what will happen. And like I said, I had two. It was in October. Julia Thévenon sent me a mail, and then last week or 2 weeks ago, it was Pascal Joseph from Switzerland. Yeah, I have two new cases, two new variants, and based on where the variants are, I do not dare to make a prediction whether they are pathogenic or not. So I cannot include them in the paper because I'm not sure. And if I have to make it again, yeah, okay, we'll face another delay of 3-4 months. So,
Yeah.
and that's
Well,
how it always got delayed, right? But now we
yeah.
have at least 27 cases that and
So
so sometimes the information is just in the gene matcher database. It is a very nice thing that exists amongst the human genetics community. So the people get kind of whole exome sequencing or so, and this variant comes out, and the clinician just puts it in the database and looks if there are other reported cases like that, and then get in touch. But then it always comes down on finding somebody who can really test the pathogenicity of the mutations. And often that person is just not available, or nobody knows who can do it. For the PP2 genes, yeah, the expertise is there, and yeah, it can be done, but yeah, somebody has to do it, and there is no information about, just information about the clinician who submitted that information. And the variant, and if you're lucky, a little bit of symptoms, but sometimes it's just a boy with a developmental delay, and that's it. So it really depends, yeah. So no, the information on the genetic report, nothing. So you have to contact the people, and then some reply, some never reply. And then, yeah, okay, there is a potential case out there, but yeah, if there is no contact, yeah, then you cannot include it.
Yeah, and what we've done in the past, Sharon, is like we've contacted main genetic testing places and saying like, there's a new publication on that. So let's do that this time too. Once it's out, we can go back and say, hey, if you had any, patients that you've done testing for where that's flagged, here's new data. It doesn't always lead to anywhere, but at least they're informed. Yeah.
Mhm.
Any other questions? I want to be sensible to everyone's time. We have another 15 minutes, so I'm not sure what else you had in the presentation, really, but
Not so much, but maybe this is kind of important also to know, that shows like the similarities between the four genes. But I actually already touched upon it. So the number of cases, and like R5C and R5D being the most related, I think, but with some minor differences, like the epilepsy that is a little bit more frequently occurring in the reported cohort, and then the let's say the developmental delay is in general on the milder end for R5C compared to R5D. For the rest, like, the macrocephaly is in common. The hypotonia, where is it? It's also in common. It's in common for all the genes. And yeah, for the rest, there's nothing really significant. So it's, so we propose to call it type 4. And so with R5D being type 1, R1A type 2, RCA type 3, and then R5C type 4. And then in the summer, there is like a fifth gene. It was only one patient, R5E. So we have R5C, R5D, and R5E. That also looked, I mean, it's only one patient, but it was a boy from Sicily, and he had also a same mutation, the same place. It was also an E2K mutation that we found that is present in R5D and in R5C. So based on the conservation of that location, and although it is only one single case, I think, and we tested also it in our assays, and it also showed like AC binding effect. So we think it is also pathogenic, and that could be our type 5 already. And it might not even stop there. So, I mean, and that's still, diagnosis, only diagnosis. Then we're not even talking about understanding what the cause is, or going to a treatment that acts on that cause. So, but okay.
yeah. Thank you. That's all very, very helpful. Just want to keep a few minutes for questions, and then we can continue if there's any questions. If they're specific to your family, that's also okay. Whatever you have on mind, you can
Could I ask a question if that's
Yeah.
okay, Carol? Yeah, so we've in Alfie's case, I would say he has been quite significantly affected. So just trying to kind of understand, I guess what we're still not fully clear on is that down, do we think the severity of his symptoms are linked to how mutated the gene is? Is that right? Or is that still kind of not quite clear?
I don't know what his specific variant is, but usually, yeah, there is a kind of link between the type of his variant and then I mean where in the protein the mutation occurs, because the type of variants, it's the same for everyone. It is just one building block that is changed to a wrong building block.
Okay.
That is for all the cases, almost all the cases like that. But then it depends on where in the protein this change occurs. And for R5C, I think we can say if the change occurs in this orange region, in the acidic loop, then it tends to be more severe. But there are exceptions. So some places it is not the case. But a real correlation, we haven't really been able to see it. For R5D, it's different. So there we really know, like some variants, they're usually associated with a more severe presentation than others. For R5C, it's more diverse, it seems. So for those cases where we have that are affected, let's say by the same variant, and it's not so many, yeah, it's less clear. So it's one. Let's say if we have two cases affected by the same variant, we have one case that is more mildly affected and one case that is more severely affected. Yeah. So what do you conclude then? Is it a more severe variant or a less severe variant? So, but this is, yeah, I mean, this is not abnormal. So it's always like a spectrum of severities that you see. And with children having the same variant, sometimes doing really well or sometimes doing really not so well. But usually there is like a trend. The more cases you have, you can say, okay, this tends to be more severe, but it doesn't apply to all the cases necessarily.
I think the case we've had with Alfie as well is his epilepsy has been awful, really, since he was born, and I think that's kind of always been our biggest concern, that's probably possibly played like a big part in the structural changes he has on his brain, like on his MRI, and also probably with his development as well. So he actually had a VNS fitted just 2 weeks ago. So that kind of shows you where we've got with his, we were like multiple AEDs, ketogenic diet, and now we've had a VNS done 2 weeks ago. So really hard to treat, really hard to control seizures.
Yeah, I think in my mind, from these 27 cases that we have seen and looked at in detail, I think that is the most the biggest problem, I think, the epilepsy.
Yeah.
And it seems to occur more frequently compared to what we see in R5D. I don't know why. But yeah, yeah. So maybe the developmental delay is milder, but the epilepsy is more frequent and sometimes refractory. And you said try the ketogenic diet.
Yes, we're second time round on keto. So Alfie had his seizures started right from birth. So we have videos of him straight after delivery having seizures. And then, so when he was about 6 months old, he went on to keto. And that worked really well.
Yeah.
When I say really well, I mean it stopped. We were never seizure free, but it stopped the hospital admissions, and he was only needing like buckle maybe twice a year. Um, we then did like a slow wean off keto because we'd been on it for over 2.5 years. Things were relatively stable. His EEGs were looking good. And then he weaned off quite well, but then within 48 hours, we were back in hospital. He had a really, really big seizure at home. And then for about a year, we just really battled, and it just really, he was just really bad off the diet, and it was kind of escalating, and it was ending up with he had to be intubated twice. We had like a PICU admission with seizures. So we went back onto the diet, and we've kind of been like, I guess, slowly like clawing our way back to try and get some level of control. But he is definitely better on keto than he is off keto. So we're a year now without any hospital admissions linked to his seizures since we've kind of been back on the diet.
Yeah.
He is also optimized. He's on 3 anti-epileptics. He's on Keppra, clobazam, and carbamazepine, and they're all fairly really well optimized. So I think it's definitely a combination of like keto and the medication that has worked best for him. But we've still been struggling with prolonged seizures. So I think he's had to have buckle about 8 times this year. So that's why we've now obviously gone down the VNS route, and we had the VNS put in. It's still really early days. We only had it done 2 weeks ago. So but we're very hopeful that that's gonna really kind of help him. But I would say that's definitely been the worst part of his condition. I would say is the seizures, and trying to kind of, because it just affects everything else. That's that's like in terms of family life and quality of life, and so
Yeah, I asked specifically about the ketogenic diet because I heard many good things about this with many families, and often with refractory epilepsy, where the ketogenic diet was like a miracle solution. So I think for everyone else, I mean, it is something that you really have to keep in mind, I think, if it would come to that point that I mean that it is a really viable option to treat or to prevent the seizures. Although not every neurologist is such a big fan of this, I think, but as a parent, I think you have to know that it can be of a big value here. On the other hand, I also have to say, research-wise, we haven't looked a lot into the seizures or into the, like what the cause is or how we could treat it. It's also so variable, huh? So even within children with the same variant, some experience the seizures from birth, like Alfie, and they are really bad and difficult to treat. Some never ever experience them, and also the kind of seizures that the children have, they are so diverse, and so there is nothing, it's difficult, and research-wise, it has not been looked into. None of us have actually really looked into it. We know in the mouse models they can be recapitulated for R5D and for R1A. So likely if we would have a mouse model for R5C, we would be able to recapitulate it. And then we could study it better, of course. But I've always felt from the very beginning that this is something that affects the children and the families most. That is the seizure, or the fear of when will it occur? Will it occur ever, or and when will it occur? With my child, And I don't think we have any answers or we can provide you with any clear solutions. Only, like I said, the ketogenic diets, I hear stories over and over and over again that it can be really, really beneficial with our children.
Yeah. And it's even, I mean, like selfie, it's
And
not,
now again, your story kind of,
yeah, it hasn't stopped the seizures, but it's so interesting because that year when he came off, the seizures looked the same, but they were just less intense. So the only way I could describe it to the doctors is like the fist clenching or the jaw clenching, or like they would still be there and they are still there, but they're kind of just like not as intense. So it kind of almost makes the seizure itself almost like this less severe. And he responds better to buckle when he's on keto. So when he was off keto, we went through this horrible period of where he just wasn't responding to the buckle at home, and that's why we were ending up kind of being taken into hospital. But once we got him back on keto, he just started responding to it again. It's and it's really, I don't think even our neurologist can't kind of explain a lot kind of how it works, but it seems to have this very, I mean, when he was a baby and he went on it, it kind of really calmed him down. Like it calmed the sensory issues down as well. And he had these kind of also, he had like a kind of a movement disorder as well. And that kind of almost disappeared kind of once we got him onto keto. So there was definitely something, very calming about, I guess, the brain using fat for energy instead of glucose. Kind of,
but that's true. I mean, and it also makes sense in terms of what we know that the mutations really affect and the pathways that the mutations affect in the brain. So that there is this link with, like metabolism or and specifically glucose metabolism. So some of the genes that regulate that are probably affected by the mutation. So I just think, we need to do a bigger effort in the research team to look into that, I think, with the models that we have. But for R5C, we don't have a model yet. So I realized that very well. And so what I would like to propose, and I mean, I've taken the initiative already, is to try to engineer, I would say, the two most common variants. This is also how we pick the variants for the other genes too, because you cannot make a model for 20 different variants. You always have to pick one or two. But to engineer them in the iPSC allelic series that we made, actually, I think that would be the most, economical and quick way to include an R5C model in the research, I think. So we started to use these iPSCs now for the R1A studies. So from the allelic series that was made by the research team, and start to differentiate them to neurons in a dish and even brains in a dish, like brain organoids, and it works very well, and it's quick, and it's maybe also not very cheap to do, but cheaper than mice and quicker than mice. And it would also allow us to have a model that can be used for screening drugs and so on, and even model the epilepsy. And so, I mean, I spoke with Kyle about it, so from UC Davis, and he was willing to help us to make these mutations in the iPSCs. But in the meantime, I haven't heard anything about it anymore. I hope that it is ongoing, but I haven't heard anything about that. But that would be, I think, a way, because we have to have some model, a readout where we can measure the epilepsy. And in the mice models that we have for R1A and R5D, we can see it. The mice also get seizures. But for R5C, we don't have a model yet. So I think, yeah, I mean, if we really want to include this gene also fully in the research, then we have to invest in a model. And then I think to add that to the iPSCs, that would be, so that is stem cells then. Yeah, sorry I have to explain a little bit better, but it's a kind of stem cell. And we have R5D mutants in there, R1A mutants in there. And it would also allow us to compare the three together in that model. And so you can differentiate these stem cells into neurons, and you can measure them. And epilepsy is usually caused by an overexcitation of the neurons. So the neurons fire too frequently and too high, and you can measure that by electrophysiological measurements. And I mean, that would be perfectly possible to achieve in this model. And I would even say these stem cells and even the brain organoids that you can generate from them, they are the best model to look at epilepsy, specifically at epilepsy.
Yeah.
Better than the mice, because in the mice you have to start, you know, can you imagine how you have to do in EEG in the mice? I mean, you can never do it with 100 electrodes as it is done in your children. It's usually only 2 electrodes, and where do you have to implant them? How do you have to interpret that EEG? I mean, it's so many different, so it's so different. And I mean, at UC Davis, there is Jill Silverman who has some experience to measure it, but I don't know.
So, I mean, really, this is in line with kind of the process for the other genes too, right? We did like the publication, then we did the cell models, mouse models, same for 2R1A. So that's kind of just like the natural progression here as well, you know. So we'll definitely continue forward. And Simon Searchlight does have 2R5C samples as well from the families that attended, so they can create our iPSCs as well.
So that was going to be my next question. So there is blood samples being taken.
Right, Sharon and Min, right?
And they are in Simon's. They are in Simon's, but then we still
So they'll
the
create them for us
are
if
not
we
being generated, but because
let's look back. Let's look back on that and see, you know, the next step now that the publication is almost done, and then we'll see the best way to enable that work moving forward. Let's see.
But I would be in favor to introduce it in the allelic series, because then we can,
and I think I'm gonna go back and see what's included in that. I think we do have other models, but I'll circle back on that. Let's see. Hi, everyone. Nice to see you all. Hi, Alan. Hi, Nicolo.
Nico, say ciao.
You're tired. Ciao.
Yes, he's tired in the nose. Thank you so much. Bye. Bye.
Let's see. Min or Rachel, do you have any questions?
Attends. Oui, non, ça, non, là qui me viennent à l'esprit, là maintenant, non, mais peut-être plus tard. Mais merci beaucoup. C'était très intéressant.
Great. Min, anything from you?
So, we are going to meet the Alan's neurologist early next year. So with this, you know, I know that your publications still need to be, you know, in revision. So, but we should expect to come out, you know,
this year,
first quarter of the year, or maybe later on.
I expect to be able to send it back Friday. So we see the statistician on Thursday, and then we should be able to send it back on Friday or in the weekend. And then, yeah, it will have to go back to the reviewer. He has to read it, has to give his consent. Then it goes back to the editor. Then the editor has to give his consent. So I think, yeah, maybe in, let's say, 3 weeks' time, we should have the accept decision normally. I have to touch
No,
wood
we have no control on that.
because
Yeah.
really it should have been like Wednesday. I was expecting, okay, it's the accept, and then I said no.
No, but I
Like the
think
stupid statistical test that we did for all the other papers was never a problem, and yeah, okay.
But
So
I thought it's take longer because over the summer, we're at the conference. I thought it's gonna be a lot longer, but then now, when we meet today, it's great to hear that you're already pushed forward, and
yeah. Well, I promised at the conference that we would submit it soon, and we submitted it in August, and then it's just, that's how it goes. It takes a month at least before you get the comments. Then we had the revision, then we submitted it again.
So
And then
hopefully it'll be out this year. But Min, if before your appointment it's still not out and you think there's any information that you need, just reach out. We'll provide that, if you think any of the information that Verily just presented will be helpful. I mean, I'm crossing all my fingers that it'll be out before,
So the
the end of the
neurologist,
year, but we can provide that information.
is
Go ahead,
he on
Verily.
the paper, then your neurologist? Is he on the paper as a co-author?
I just read through the name. I recognize the older the one of the previous genetic counselor that Alan first met when we got the genetic test, and then I think I'm gonna, because there's so many names. I only recognize one name. I don't know if my other because he has a new neurologist lately, because there's a change, and then the older one, the original one, she moved. So
I also don't know by heart. It's not Rebecca Levy, right? Rebecca Levy? No.
Oh yeah, Rebecca.
Rebecca, yeah. She's on the paper.
Oh, okay.
But she's not your neurologist. No.
She's the new one that's just assigned to Alan, lately.
Okay.
So yeah.
She has been really helpful, but and responsive.
Oh, she is. Yeah. We only able to only meet her like once a year, because we usually see the nurse practitioner, and then but I'm excited to see her in person, the next year, early next year, so then we can kind of, and then she's really good at reading all those research paper, all the things that she's really good about, reading all the scientific,
but she's she's a good scientist also, I think. That's why.
Sharon,
Not
anything?
just a clinician.
I don't have any questions. I just want to do a follow-up to Lindsay, that just to let you know that our son has a very similar epilepsy story to yours. So maybe we could connect to talk. But Emmett is 15 and he was on keto for 14 years because we pulled it off and on. And the second we would pull it off, we saw instant seizures do this. And so I know it was a long time, and a lot of people would say, we're unsure of like how that would be being on that long, but he grew fine. Like we never had any nutrition issues. We just kept up with the dietitian that whole time. We did labs frequently to make sure we're not missing any, key parts of his diet. And anyway, so if that's helpful to you, it definitely helped him. And then we've also had to do the VNS and have experimented with all kinds of meds, and right now we're probably at our best place we've ever been with the combo that we're on. So I don't know if you wanted to chat later about
yeah, that would be so helpful. And I mean, I think from a similar thing, like Alfie has always actually eaten really well, so even on keto he eats really well, and so and again his bloods are fine, like from they check all like the minerals and make sure that he is getting kind of what he needs. But yeah, I was just And how have you found the VNS? Have you found that to be helpful with regards to the seizures?
We have, yeah. He had it for 5.5 years before we hit the battery. We needed to replace it. And so he just went and had it replaced this last year. And so we liked it enough to say, hey, let's, do it
Yeah.
again. So, yeah, it just makes the seizures for Emmett just a lot less severe. Like he still has a few, but they're way more mild than they used to be. So we
Yeah.
like the VNS for that.
Yeah. So, and I think that's kind of it's really just we accept very much that Alfie will never be seizure free. So it's just about trying to make the gaps between the big seizures as long as possible.
Yep.
And so when he does have the big ones, just trying to keep him at home and just kind of improve his quality of life. We had an awful year. Last year was just the absolute worst. We were just constantly in and out of hospital. And I think as a result of that, I don't think actually I will take him off keto again. I think I might be like you and we'll just keep him on it because he is so much better on it than without it. And it kind of really unveiled the beast of his epilepsy with him not being on it. In the first 6 months of his life were exactly the same. When he wasn't on keto, we were lucky that we were able to get him on keto at such a young age. And he took to it really well, and he enjoys his food. He loves his food. And actually, I think parents worry about it being kind of like time consuming in terms of like the preparation, and it is, but I would much rather do that than be in hospital.
Yeah, for
So,
sure.
I'm absolutely more than happy to kind of take that hit, and I feel that actually you just get into the swing of it and it just becomes like a normal part of life, doing the food prep and everything for him. So he's on a very soft diet as well. He can't really cope with much texture, so we kind of have to blend everything as well, but that makes things a little bit more tricky. But generally it, I've seen, I think having that period of time with him off the diet, it was just so awful that I just don't want him to go through that again. So but yeah, no, it would be really helpful for us maybe to chat privately or something about your kind of experiences and things. So
I'll connect with you over. Are you on Messenger?
Yes, we are. Yes.
Okay. Yeah, I'll find you and send you a message.
Yeah, that would be great. Thank you so much. Thank you.
So a couple questions for the group, just in line with that. I mean, Sharon and I have talked back and forth about if it makes sense to create like a subgroup, even in like a WhatsApp group or some way to connect the 2R5C community together. And the reason we've been reluctant is like, one, we don't want the sub community to feel like segregated, but also like, how do we handle languages, right? Because if we have different language speakers, like our ambassadors do a good job supporting the languages. But if this group thinks there's value or just even emotional support to be part of like at least like a text message or some way to be able to contact each other, we're happy to get that started. Any input or strong feelings either way on this?
I don't have any strong feelings either way. I think for me, actually, just from just understanding the research a little bit more and also the comparison, it seems like and even when I speak to a lot of the 5D families, there seems to be such a crossover anyway. So it has been amazing, I think, for me, definitely to be able to connect with some 5C families. But again, the spectrum is so wide that actually even some of the other 5C families I've connected with, their kind of experiences is still quite different to mine anyway. So I don't mind either way. I'm happy kind of either way. But I find just the main Facebook group itself is just so helpful. If there's anything I want to find out or clarify, I can just put a post on there and people respond. And
yeah, and they're so similar. So I do think, we benefit from each other's communities. So here's what I'm going to ask everyone. I'm going to put in the chat this form, which is like our database. even if you filled it before, please go ahead and do it. At least we'll collect like the emails of the 2R5C community, and we can just send one email out. So if at any point we need to contact just something specific to 2R5C, like this meeting as an example, we'll have a way to do that. So I would propose that, and then if as the community grows, if we feel differently, we can go from that. Does that sound okay, like as a next step?
Yes, yeah, that sounds fine. Yeah.
Okay. So please do click that form, is like literally just your name, your email, like just so we have access to everyone. And then we'll circulate that later to make sure we have that as well. Last housekeeping, Simon Searchlight, if you're not on there, please take the time to do that. We're really itching to get to that 10 families on Simon Searchlight so we can start getting these updates regularly on the community and how we're doing. So if you have any questions on that, Sharon, you have something to add?
I just was gonna say, just to say the same thing and say, please do it. Because last time we looked, there were only 5 that went through the whole process for C. Did that change to 8, or they're just 8 that are in there somewhere?
I'll run the numbers again, Sharon. We have there's like Simon's now created like this way for us to look up these numbers, so we don't have to like
Okay.
keep going for them. So we
Yeah.
can check every day if we want to, and we can look per gene, per region. It's really nice. So let's do that. I mean, we circulated that in the last newsletter, I think, but I'll go back and look at it. It's hard to keep track of
Like
everything.
how many went through all the way. Yeah, and just to reiterate, if you have any issues, to reach out to one of us, all of us. But
yeah, and they support
languages.
languages. Yeah, French is definitely on there if that's needed. Okay, sorry we went over time, but I hope that was helpful for everyone. Verily, thank you so much as always for your dedication to really support our community. We're committed to keep moving through the process and finding more, as we learn from one gene. The hope is to be able to check, does that apply to the other genes, and expedite that process going forward. As always, if you have anything, please do reach out. We're available to answer any questions. Really, anything else from you before we wrap up?
No, I hope I will be able to bring the good news very soon about the acceptance of the paper. That's all.
Yes, I think it'll come. We're here waiting for it. So, yeah, otherwise, I mean, I do have a couple action items here, so we'll follow up on that. But let's do that at least once a year or more if there's a need for that, even without, the presentation, just getting together and talking and sharing experiences. I think that'll be good. Anything else from anyone?
Hopefully, the next time meeting we have see more families. I was excited.
Yeah.
I joined. I talked to Sharon, Rachel, and another family. Yeah, and then another family in Italy. But then the rest, like, I know the numbers growing, but like, where are they?
Yeah.
I was hoping like once we get the publication out, then that will probably help to grow numbers. Yeah. Because I felt like I felt we had such a good geneticist, because I don't know if you could, I know, sorry, everyone probably wants to go, but Alfie actually had like trio exome done when he was only a few months old, and it just came back as like that they couldn't find anything. But we actually moved. We were living in London at the time, and then we moved out and moved to Hampshire, and then we obviously then got referred to the kind of the genetics team like locally to us. And I must say that the geneticist we have now, she is absolutely phenomenal. And just how you were explaining about like the difficulty. So she knew about 5D. And again, we then had whole exome sequencing done. And that's when, again, the report came back saying that they couldn't find anything. But actually, because she was aware of the 5D, when she was looking through, I think, the report, she saw the 5C and thought, oh, there could be something in that. And I think that's when she got in contact with you. And then actually we were expecting it to be quite a long drawn out process, because she said that there was only one other child that was ever reported or something, which was probably that one that you were kind of talking about. But
I haven't been able to track him back though. But it was in the UK. It was in the UK, a boy.
That's what she said. Yeah. And we were like really, she was kind of really preparing us for this kind of very long, like kind of process really. And but then obviously then we found like Jordan's Guardian Angels, and then obviously she's kind of, and then I don't think she realized when I actually found Jordan's Guardian Angels online when I started doing my kind of Googling and things like that. And but yeah, it's I wonder if we hadn't had her as a geneticist, would that have even been, because like if nothing is published yet, it's only when it gets published, isn't it, that they're able to
that's why it is so important to have it out. I think that I mean, and in a good journal, so that the exposure is broad, and I think it cannot it will lead, it cannot be different, it will lead to more diagnosis. That's I mean,
because I'm right in
thinking
a certainty. It would have, when we had the exome done, it would have shown on the exome, wouldn't it? Am I right?
Yes, it should have, but then it would probably come out as a likely pathogenic or a variant of unknown significance or something like that. So now, I mean, if the symptoms fit, even if it would be a variant that is not reported in the paper, I mean, it will likely be given as a positive diagnosis, and then if it is a new variant, then I will get a mail and say it's a new variant. Can you test it? And then like that, the diagnosis can be confirmed. But if the gene matches already, that is the first step, of course.
Yeah, because here in the UK, I mean, technically Alfie doesn't really have an official, I mean, our geneticist has put it for us in a letter, but the labs, so like the National Health Service here, they won't confirm the diagnosis until the publication is out, which actually it doesn't for us, that it doesn't, you know, Alfie gets all the support and services he needs because it's based on need, not necessarily a diagnosis. But yeah, so I do believe the numbers are gonna, I think once we get this publication out, then I think the numbers will get much bigger. Yeah, I'm sure.
I expect that. But yeah, we'll have to see.
Yes. Yeah.
So your geneticist, is it a Helen? No. The name of her?
It's Lachlan. I think it's Catherine.
Yeah. Okay.
Yes.
Catherine.
Yes. Yeah. She is I think it comes under like Wessex, even though we're Hampshire genetics.
Yeah, it's
services.
possible.
So
but yes, I have been in contact with her.
Yeah, very good, very thorough. And like I said, when she explained it to us, she actually, I think, you know, heard of 5D, and that's kind of, I think, when she was then going through and saw the 5C, she thought, okay, could that be could that be something?
But so also to solve that problem, I think, I mean, even if they see another PPP2 gene, they have to nobody makes the link. So that's why it was also important, you know, to change the official name of these genes, and so that's because now it has this, it's like a PP2A syndrome with different subtypes, and also for that I think so we are also writing a paper on that, and a kind of overview paper to make that clear, and so that's also 80% finished. Because a genetician just looks at the gene and doesn't make the connotation that some genes are kind of similar or linked, but obviously Dr. Laughlin made that connection.
Yeah, and I think it was like from the conversation I remember, it was almost like a bit of a, not a fluke, but she'd done either some reading or she'd been to some presentation or she'd heard something about PPP2R5D. And so then when she got Alfie's report and she saw the 5C on there, she thought, oh, and then obviously she remembered like that it was kind of similar, kind of the presentation was quite similar. And that's kind of then what made her kind of start looking into that. Whereas I think maybe the geneticists we had when we had the exome done maybe didn't have that kind of knowledge. And so it was just kind of overlooked, I think, really.
Yeah, probably.
Yeah.
Probably.
So yeah, so
Yeah.
I think you know, we potentially, but again, you know, so it would have been 2019 when he had the exome done. And I think it was probably, I can't actually remember. He was about 2, I think, when we got the, it was maybe like 21, 22, something like that, I think, when we got the whole genome sequencing done. So, but yeah, I remember when I was reading up on it that I thought, oh, I bet it was actually there when we had the exome done, but it just wasn't, because it wasn't known about. Yeah, yeah. So, but you know, we're at where we are. So
yeah. Okay.
All right. Well, thank you, everyone. Yeah.
yay.
Go ahead, Min.
Thank you.
Okay.
Hope this see everyone soon.
Sounds good. Nice to see you all. Have a great day, and we'll be in touch. Bye-bye.
Yeah.
Bye. Bye.